The viruses used in vaccines are produced in cell lines because production in a test tube is inefficient and too costly. Currently, both animal and human fetal cell lines are used by vaccine manufacturers. Production of viruses in cell lines results in residual DNA and cellular debris from the manufacturing cell line in the final product. For instance, as can be found in Merck's summary basis for approval, which is Appendix F, page 3, for the chickenpox vaccine, over 2 micrograms of residual double-stranded human fetal DNA are present in each vaccine, which is approximately twice the amount of the active ingredient of the vaccine, which is the varicella virus, a DNA virus. As FDA scientists state in their 2008 publication in the journal Biologicals, the danger of residual DNA in vaccines has been debated for over 50 years without appropriate studies. The 2008 publication by these FDA scientists demonstrates the real and present danger of human DNA residuals in vaccines, which include cancer, autoimmunity, and genomic disruption. As I have already mentioned, the IACC strategic plan well summarizes data demonstrating de novo genomic disruptions at varied sites in at least 10% of children diagnosed with ASD. Having worked in commercial biotechnology and clinical development programs, I was aware of the residuals that would be found in vaccines, and having also worked with homologous recombination and molecular biology, I was also aware that the human fetal DNA introduced in vaccines has the potential to elicit autoimmune responses or to incorporate into the recipient's own genes and disrupt normal protein production. Inflection points, change points, are clear by eye in U.S. autism prevalence data from the Department of Education, as well as from the California Department of Developmental Services. This intriguing and perplexing visual assessment of U.S. autism prevalence, together with publications on autism rates in the UK, led Sound Choice to more fully investigate. What we have found is that across continents and across decades, changes in autism disorder, not considering autism spectrum, but only autism disorder, are clearly associated with the introduction of vaccines produced using human fetal cell lines. Each time we inject our children with one of these vaccines, we are also injecting them with residual fetal human DNA. So not only was human fetal DNA introduced to our children with the MMR2 and chickenpox vaccines, but a retrovirus capable of enhancing genomic insertion was also introduced in these vaccines. Human endogenous retrovirus K is present in the MMR2, in Merovax, which is a monovalent rubella no longer produced, and the chickenpox vaccines. This work was published in the Journal of Virology in 2010. HERV-K, human endogenous retrovirus K, is a virus related to the MMLV virus, a virus that was used to deliver gene therapy in clinical trials for boys with SCID disease, that's severe combined immune deficiency. They're also known as the bubble boys. The MMLV virus caused inappropriate gene insertion and subsequent somatic gene mutations in 4 of 9 boys that led to the development of cancer, although experts testifying prior to the initiation of those clinical trials estimated the risk at 10 to the minus 26. The actual risk turned out to be 4 of 9 boys developed cancer. These studies are published and are included in the appendix of the binders that I have with me. I also need to say that it is important that independent groups conduct these types of analysis, and several groups should independently examine each question so that children are finally helped and protected. One of the key scientists funded by the CDC to do studies into MMR and autism links, studies used to dismiss the MMR autism hypothesis, was indicted on April 13th this year, 2011, on 13 counts of fraud and 9 counts of money laundering. Furthermore, Dr. Thorson had already been cited for academic misconduct by his own university and was charged with embezzling $1 million grant from the Centers for Disease Control.